Assalamu alaikum. Today we will start our second online lesson, Prokaryotes. By the end of this lesson, I expect from you to be able to explain how prokaryotes are classified, to describe how prokaryotes vary in their structure and function. You can also depict the role of prokaryotes in the living world, to describe how bacteria cause disease, and to define what are emerging diseases. In front of you, you have a diagram of both eukaryotic cells and prokaryotic cells. So it's just for recalling your information from before. We need to remember that eukaryotic cells are having membrane-bounded organelles and membrane-bounded nucleus, while prokaryotes, they don't. Eukaryotes and prokaryotic Venn diagram is showing the main differences between eukaryotes and prokaryotes. Now, as we said, the main difference that the membrane-bounded nucleus and the membrane-bounded organelles that are found in eukaryotic cells only. Now, eukaryotes could be unicellular or multicellulars, while prokaryotes, they are only unicellular. Both types of cells, they have a cell membrane, ribosomes to make proteins, and of course, the cytoplasm. Here we can see a typical bacterial cell in front of you. Now, as we said, all prokaryotes are actually unicellular organisms. They have no nucleus. If we look at the diagram, the cytoplasm is containing the DNA without a nucleus. Prokaryotes include two main types of organisms, bacteria and archaea. If we look here at this diagram, we can see, let's start from the plasma membrane, the cell membrane here. Above the cell membrane, there is a cell wall, then a capsule. We have pilus. Pilus helps the bacteria or the prokaryote to cling to surfaces. There is a flagellum that helps in movement. If we will go inside the prokaryote cell or prokaryotic cell, we will find the purple is the DNA, and we have the cytoplasm. Inside the cytoplasm, there are ribosomes that they are responsible for manufacturing proteins inside the cells. As we said, prokaryotes are having two main types, archaea and bacteria, or we can call it eubacteria. Now, the main difference between both of them, that archaea is capable of living in a very extreme conditions like a salty environment or a very hot temperature environment, or even an environment that is rich in methane gas. They have different chemical composition than eubacteria, that is different composition for their cell walls and cell membranes. And also they have different DNA sequence. Eubacteria or bacteria, it can be found everywhere around us, on us, or even inside us. Some of them are helpful and we are using it to make our food, vitamins, or nutrients. Some other bacteria is harmful and it can cause diseases. Prokaryotes are not the same. They can vary or they are different in sizes. They do have different sizes, different shapes, and also their movement is different. If we look at these three pictures, we have different shapes of prokaryotes. This is the bacilli shape that looks like a rod. This is the cocci or spherical shape. This is a spirella shape that is a spiral. It has a spiral shape. For movement, some prokaryotes, they cannot move. They are not capable to move. Some of them, they use a flagella to move, and some of them, they can glide on secretions only. I want to recall with you when we talked before in term 1 about autotrophs and heterotrophs. Just to remember that autotrophs are organisms that are capable of making their own food, while heterotrophs, they cannot make their own food, so they need another organism to obtain food from them. They depend on other organisms. Talking about prokaryotes, also, some of them are autotrophs, some of them are heterotrophs. The energy captured by prokaryotes could be, like we have here, the heterotrophs, and these three are autotrophs. Heterotroph bacteria, or prokaryote, they take in organic molecules from the environment or other organisms, so they depend on other organisms to obtain energy. These three types, all of them are autotrophs, so they are capable of making their own food, but we have the photoheterotroph. Photoheterotroph, they can so they can be heterotrophs, and at the same time they can be autotroph. So it's based on the conditions that they are living in. So sometimes they can be heterotrophs, they need other organisms to feed on, and sometimes they can be phototrophs. It means that they need light energy to make their own food. The second type of autotroph prokaryotes that are called photoautotrophs, they use light energy to convert carbon dioxide into carbon compounds that they can use it as energy source. The last type of these autotrophs is the chemoautotrophs. So they are using the chemical energy to make a compound that help them to take energy. So they are making their food using chemical energy. Some prokaryotes, they need oxygen to release their energy. Some of them, they don't require oxygen to release their energy. Now, these are different prokaryotes based on whether they require energy or they don't require, whether they require oxygen or they don't require oxygen to release their energy. Now, obligate aerobic prokaryotes, they require oxygen, so they are depending on cellular respiration, so they need oxygen, they cannot live without oxygen. The second type of prokaryotes, they are called obligate anaerobic, so they require a lack of oxygen, so they don't need oxygen, and they undergo fermentation. They might die in presence of oxygen. The last one are the facultative anaerobic, that they survive without oxygen, but also if oxygen is present, they will not die like the obligate anaerobic. They can be either using cellular respiration or fermentation. Prokaryotes reproduce using binary fission. Binary fission, which is the division into half, is a kind of asexual reproduction. It's the most common from reproduction in prokaryotes, the most common form of reproduction in prokaryotes such as bacteria. In this process, the cell replicates, as you can see in front of you in this diagram. The cell will start at the beginning to replicate its DNA. Here we have 2 copies of the DNA. 2 copies of the DNA here. So this is 1 copy, and this is the second copy, of course, an identical copy. Then it will start to divide into half to produce 2 identical cells by the end. Sometimes the conditions might become unfavorable for prokaryotes. So what they are doing, that many prokaryotic cells form an endospore. Endospore is a thick internal wall that encloses the DNA and a portion of the cytoplasm. Endospores can remain dormant for months or years, waiting for good conditions. This diagram is showing an endospore, and this picture here in front of you is a real picture under microscope for endospores. These green structures are the endospores within the bacterial cell. If you notice, here you have links for related to this slide. So whenever you find this link, please go to it. It explains more about the slide that we're talking about. So here you will find that it's explaining about the endospores, and the previous one you have animation for binary fission. So please use these links to understand more. Conjugation is the process that prokaryotes use it to exchange the genetic information. So it's like a sort of way that prokaryotes are making genetic variations or genetic differences. This is a picture here. This picture is a real picture under microscope for 3 bacterial cells, and they are undergoing the process of conjugation. This is here. This is clarifying how conjugation is happening. These are 2 cells, 2 bacterial cells, 2 prokaryotic cells. So there will be a conjugation tube between the 2 cells. A part of the DNA of this cell will be copied first and transferred into the second cell, and this will give a genetic variation for the prokaryotes. Also, don't forget that you have also a link for animation of conjugation. Prokaryotes in our environment. Prokaryotes play important roles in the environment as decomposers, producers, and nitrogen fixers. We know, if you remember, that decomposers are the organisms that they can recycle or return the main raw materials into the environment, back to the environment. Food chains everywhere are dependent upon prokaryotes as producers of food and biomass. Nitrogen-fixing bacteria and archaea provide 90% of atmospheric nitrogen. If you remember also the nitrogen cycle, so nitrogen fixation is basically depending on bacteria or a nitrogen-fixing bacteria. Of course, not all prokaryotes are helpful. Some prokaryotes are harmful and they can cause diseases. Like here in this table, we have some examples of diseases that are caused by bacteria, and it's mainly caused for human. So like some examples like Lyme disease, tetanus, tuberculosis, bacterial meningitis, and strep throat. To control bacteria around us, we have some methods like a physical removal of the bacteria using disinfectants, food storage, food processing, and sterilization by heat. To prevent bacterial diseases, scientists developed some vaccines. Vaccines are a preparation of weakened or killed pathogens or inactivated toxins that they will release antibodies inside your body that it will fight the disease or the bacteria. By the end of our lesson, we will talk about something that actually we are living it nowadays, which is called emerging diseases. Emerging disease is a previously unknown disease that appears in a population for the first time, or a well-known disease that is suddenly becomes harder to control. In recent years, new diseases, this map is showing some new diseases such as Zika and Ebola, they have appeared. At the same time, some diseases throughout to be under control have come back. Both examples are classified as emerging disease. Of course, we won't forget that COVID-19 or coronavirus is a new emerging disease that we are suffering nowadays. Thank you for listening. Hope you understand, and contact me in case of any doubts or any explanation, more further explanation. Thank you for listening.